Chapter 16 is about the central dogma of biology, how DNA is read to make RNA through the process of transcription, and how the RNA is read to make protein through the process of translation. So we're going to start with transcription. Transcription and translation both have 3 steps, a beginning, a middle, and an end. So to begin transcription, we begin with initiation. The start site for transcription is the beginning of a gene, which is marked by a stretch of nucleotides called the promoter. In eukaryotes, the promoter typically contains a sequence of TA repeating, which is called the TATA box. The promoter is the binding site for proteins called transcription factors, which attach to the promoter and make a platform or a landing site for our major enzyme. The major enzyme that does transcription is called RNA polymerase II. RNA polymerase II is the enzyme that transcribes mRNA or messenger RNA that is used to make proteins. There are other RNAs, tRNA and rRNA, which are transcribed by other RNA polymerases. RNA polymerase II has 2 different enzyme activities. It's both a helicase, which can separate the 2 strands of the DNA, and a polymerase. It can read the DNA to synthesize RNA. Just like DNA synthesis, RNA synthesis occurs 5' to 3'. New RNA nucleotides are added to the 3' end of the growing RNA strand. Since the RNA is being synthesized 5' to 3', the DNA that's providing the information for building this RNA is being read 3' to 5'. So in this image, the bottom strand is the strand of DNA that's providing information to make the RNA. The bottom strand is called the template strand. It's sometimes also called the antisense strand. The other strand of the DNA double helix is actually pushed out of the way and ignored. This is called the non-template strand. Since it has the same 5' to 3' orientation as the RNA that's being synthesized, it's also called the sense strand because the nucleotides are in the same orientation and have the same letters except for the T's in the DNA on the antisense strand, on the sense strand are replaced with U's in the RNA. It's not being on top or being on bottom that makes one strand of the DNA the one that's read and the other one the one that's ignored. On an entire chromosome, there are genes on both strands of the DNA, but the genes don't overlap. So if we zoom out and we can see many genes on this chromosome, on the bottom strand there are 2 genes that are being read 3' to 5' on the DNA to make the RNA that's being synthesized 5' to 3'. So the bottom strand is being read by RNA polymerase moving towards the right. Okay. On the top strand, those 2 genes are still being read 3' to 5' and still being synthesized 5' to 3', but the direction of synthesis is to the left. RNA polymerase moves to the left along the top strand. So both strands of the chromosome are used, but at a particular location, at a particular gene, only one strand is the template and the other strand is pushed out of the way or ignored, and that's the non-template antisense strand. To finish transcription, termination occurs differently in prokaryotes versus eukaryotes. Prokaryotes have a sequence at the end of the gene called the terminator sequence, which produces a hard stop where RNA polymerase falls off of the DNA. In eukaryotes, there's a point where RNA polymerase must pass. It's called the polyadenylation signal. And once it passes the polyadenylation signal, it continues on for 20 to 30 nucleotides or so before RNA polymerase loses affinity for the DNA and eventually falls off. In eukaryotes, after the RNA has been transcribed, it has to be modified before it can leave the nucleus. And there are 3 things that have to happen to the RNA. On the 5' end, it receives a modified G cap, which is put on effectively backwards. On the 3' end, a poly A tail is added post-transcriptionally by an enzyme. And then finally, splicing occurs. The enzyme called the spliceosome cuts out the introns, or the interrupting intervening sequences, and glues together the exons, which are expressed and kept. So the final mature mRNA has a 5' cap with some spacer DNA called the UTR, or the untranslated region. The exons glued together, and then another UTR, followed by the poly A tail. These processing steps accomplish several things. First of all, a cap and a tail in splicing must occur before the mRNA can leave the nucleus. Second of all, the cap and the tail stabilize the RNA in the cytoplasm. And finally, the cap and the tail also facilitate the binding of the ribosome to allow for initiation of translation.